Hi, my name is Valerie Chamberlain. I'm a research scientist at Seacor International, and I'm based on the Caribbean island of Curacao at the Carmody Marine Research Station. So Seacor is a nonprofit organization that develops coral breeding and restoration techniques. We combine science and engineering to create new tools and technologies to scale up restoration. We also do a lot of training and mentoring for other organizations to help them develop and implement coral breeding at their own location for their own reefs. And so my role as a research scientist at Seacor is to do experiments to really better understand corals during their earliest life stages, with the goal of producing more and healthier and sturdier coral babies for outplanting on reefs. And so most corals will reproduce only once or twice a year during what we call coral spawning events. These spawning events are finely synchronized with the lunar and solar cycles and will typically happen during the summer when the temperature of the ocean is rising the fastest. And so each coral species will be spawning during a very short amount of time, let's say for about an hour, starting at a specific time after sunset, and during only a couple of nights on specific days following the full moon. And so during these spawning events, corals will be releasing their gametes into the water column, where hopefully fertilization will happen. But that's really only if all the stars are aligned and encounters between eggs and sperm are successful. However, with ongoing reef degradation, the number of potential mates on a reef has become smaller and smaller. So quite often, there's not enough colonies of the same species on one reef for eggs from one coral to have any chances of being fertilized by sperm from a different coral. So this is where practitioners and scientists like me can help. We go out on the reef on predicted spawning nights to collect sperm and eggs using special nets. And then we bring back those gametes to shore, where we proceed with in vitro fertilization, or coral IVF. And so by doing this, we can maximize fertilization success by bringing together gametes from isolated populations all in one bowl, where sperm and eggs have much higher chances of meeting each other. And then we can grow the resulting coral offspring in a culture facility, after which we outplant them back on the reef for restoration. So for many years, coral IVF has been carried out following a set of standard practices that were developed based on research since the 1980s. In general, these practices produce pretty good fertilization rates, but they're not necessarily favorable for all species. And because corals are only reproducing once or twice a year, our opportunities to do coral breeding are very limited. And so that's why it's really important to take into account these differences in IVF protocols between species, so that really we can maximize the number of coral offspring that we can produce during each spawning event with our gamete collections. And so in this study, we focused on 4 Caribbean species that are quite often propagated by coral breeding practitioners, but for which detailed information on IVF was still lacking. These 4 massive coral species included Orbicella favulata, as well as the 3-brain corals Diploria labyrinthiformis, Pseudodiploria strigosa, and Colpophilia natans. And so with each of these species, we ran 3 different experiments to establish the best IVF conditions that result in the highest fertilization success. So what we found is that in all 4 species, coral gametes remain viable for at least 4 hours after spawning. And this is really good because it gives enough time for practitioners to collect and mix gametes from multiple, perhaps distant sites, so that we can incorporate more genetic diversity in our larval cultures. We also found that fertilization in brain corals happens very, very fast, so within just 15 minutes of sperm and egg co-incubation. This is also important because it allows practitioners to rinse or to clean the fertilized eggs before they are going through fragile cell divisions. And so this reduces the risks of damaging coral embryos during their more sensitive stages of development. In contrast, Orbicella favulata requires much longer co-incubation times of 1 to 2 hours for fertilization to be optimal. And so embryos in this species really must be handled with extra care following fertilization. Finally, we've also established minimum sperm concentrations of 10 to the 5th to 10 to the 6th cells per mil in all 4 species. And this is a pretty similar benchmark as for many other coral species. So overall, this study shows that there is not one recipe that fits all. Where possible, coral breeding protocols should be tailored to each species' requirements to really ensure that we can breed as many healthy coral offspring as possible. In the next months, these findings will be distributed within Seacor's network of restoration practitioners across the whole Caribbean to really help them improve their coral breeding and restoration programs. Thank you.